ladies and gentlemen, and welcome to lecture number 6, photosynthesis. As you may recall from earlier science classes, photosynthesis is the process by which plants harness the energy of the sun to turn carbon dioxide and water into sugar. So here's the chemical formula for that. The reactants are water and carbon dioxide. You need 6 of each. And then in the presence of light energy and enzymes, you get the products of oxygen and glucose. Glucose, if you remember, is a sugar, and the plant uses this to make many other sugars, starches, and compounds in the cell. Now, the process of photosynthesis all starts with sunlight. Visible light streams out from the sun in waves. And it kind of looks like this. This is the visible light spectrum. The distance between the tops of 2 consecutive waves of light, so from here to here, or from here to here, is its wavelength. And we measure this in nanometers. You can see that red light has a very long wavelength, and over there, violet light has a very short wavelength. So, have you ever wondered, or have ever had a young son or daughter who's come up to you and said, "Why is the sky blue?" Well, the sky is blue because it absorbs all the colors except for blue, and the sky reflects the blue color back to your eyes. That's how vision works. Whatever color is being reflected is what we see. Here is the visible spectrum in the entire electromagnetic spectrum. So you can see X-rays, ultraviolet radiation, those are shorter than visible light. Visible light is just this little bitty thing in here, but this is what the plants and us depend on. And you can see it's got the nice rainbow of colors, the Roy G. Biv, red, orange, yellow, green, blue, indigo, and violet. And then the infrared or heat waves and microwaves are longer on this side. So how does the plant capture these wavelengths of light and use it for making sugar? Well, the plant uses pigments. A pigment is an organic molecule that absorbs light of a specific wavelength and uses it for photosynthesis. A pigment that absorbs violet, blue, and green light reflects yellow, orange, and red, so the pigment would look orange to us. This is an example. This would be the pigment that's found in carrots. The carotene, it looks orange and it absorbs blue, violet, and green light. The most common photosynthetic pigment is chlorophyll A. Chlorophyll A absorbs violet and red light and it appears green. This is why most trees and grasses and other plants are green, because it has chlorophyll A for its pigment. Now, if chlorophyll A was the only pigment, plants couldn't use most of the visible light spectrum. They could only use the violet and red light, and all the other colors of light would be wasted. Therefore, most, but not all, types of photosynthetic organisms have more than one type of pigment. This is why if you go inland a bit, it doesn't happen so much on the coast, but when fall happens, trees will reabsorb their chlorophyll A into the twigs to store it long term, so all that's left is the other colors, the other pigments that are normally present in the leaf. That's why in the fall trees will turn reds and oranges and yellows. This is also, if you've seen those Japanese maple trees that have, maybe that's not the right species, but the maple trees that have the dark purple leaves, those do not have chlorophyll A as their dominant pigment. They have another pigment. So how did we figure out that chlorophyll A absorbs in those wavelengths? Well, this is thanks to Theodore Engelmann's very elegant experiment, which he did back in 1882. He used a thin piece of algae, that's right here, a little algae filament, and this algae has for its dominant pigment chlorophyll A, so mostly chlorophyll A. Now he put this algae, and here's the algae right there, on a microscope slide and with some water on it, and he put some bacteria on it too. Now these bacteria need oxygen to live. They breathe in oxygen. Theodore Engelmann then put a prism between the light source and the microscope slide. So he projected this nice pretty rainbow onto the algae. And he left it for a few minutes. When he came back and looked at it, he found that the bacteria had actually moved. And instead of being just randomly distributed throughout here, there's a big clump of them over in violet, and there's a little clump in red. So he concluded that that's where the most photosynthesis was happening, in the red and in the purple lights. Pretty neat for being in the 1800s. So that's the absorption of chlorophyll A, as we said, in the purple and in the red. This shows you some other common pigments. For example, beta carotene, that makes the orange leaves in the winter, absorbs in the violet and more into the green part of the spectrum. The phycoerythroblin absorbs mainly in the yellow area of the spectrum. So you can see if a plant had all of these pigments, it could absorb in pretty much all of the wavelengths of light. Here are some cartoons of these pigments, some drawings. This is chlorophyll A here. This is beta carotene. And here are some maple trees. The ones in back, that's their normal color. And then here in the fall, they're showing their beta carotene. And here's a chart. This is just for your interest, showing you some of the other pigments and what colors they are and what they show up in. So plants have greens, oranges, reds. They even have this, I guess this anthocyanin is what gives those purple maple trees their purple color. Bacteria have a lot of different, and protists have some different pigments as well. And then the archaeans have retinol, which is interesting. Kind of weird. Okay, so now we've talked about pigments. So we can put the pigments in the plant cell. So if you look at this leaf of corn right here, you zoom into the leaf of corn, this is a cut through the leaf. There's the top of the leaf, there's the bottom of the leaf. We're looking inside. The photosynthetic cells are here kind of in the middle. You can see, and up to the top, so you can see the those little green things. This is a close-up of the little green things here. And zooming in on one little green thing, those are chloroplasts. Remember those from the last lecture? These are the site of photosynthesis. So this is the structure of the chloroplast. There are two outer membranes here. Then there's the goopy stuff in the middle called the stroma. And then these stacks here are called the thylakoid compartments. And the thylakoid's purpose is to have lots and lots of membranes. Remember from our thing on cell membranes, this means there's lots and lots of surfaces for doing chemical reactions. So now we're looking from the stroma, so we're looking from the goofy part, onto the surface of the thylakoid membrane. And you can see these things that look like coins are photosystems, and there's light harvesting complexes. So when light hits these, it excites the light harvesting complex and transfers the excitement to the photosystem. So in photosynthesis, there are two main types of reactions, which we'll talk about. The first are the light-dependent reactions, also called the light reactions. These have to take place in the daytime because they need the sunlight to happen. And this is how the plant actually gets the ATP and that sort of, and the NADPH that it needs. NADPH is a way of moving hydrogen around in the cell. So the plant will make the ATP, which is energy, and the NADPH, which is free hydrogen, and dump it into the light-independent reactions. These are also called the dark reactions, or the Kelvin-Benson cycle, and this is where carbon dioxide is made into sugar. So let's do the light reactions first. The photosystems, which were the green coin-like things in the previous display, are groups of hundreds of pigments and other molecules that work together to harvest light. Chloroplasts contain two types of photosystems, type 1 and type 2, which we'll come to in a minute. When a pigment absorbs a photon, which is a unit of light, it boosts one of the pigment's electrons to a higher energy level. So the energy comes in and goes bing, and it goes up. Normally, the electron would fall back down and the energy would be lost. However, we're in the thylakoid, so we have lots of membrane-bound proteins to pass around these electrons. Each of these proteins performs a redox reaction with the electron, and the electron releases the energy a little bit at a time. Electrons that are lost from the pigments have to be replaced from somewhere, so you can't just keep exciting the electrons. Photosystem 2 gets more electrons by pulling them off of water molecules. This breaks the water into hydrogen and oxygen. That's where we get the oxygen that's given off as a byproduct of photosynthesis, and this is the oxygen that we breathe. Splitting the water molecules like this is called photolysis. Molecules of the electron transport chain use the released energy from these redox reactions to move the hydrogen ions across the thylakoid membrane. The hydrogens can only leave the thylakoid membrane by flowing through the membrane transport protein called ATP synthase. And this makes ATP by attaching a phosphate group to ADP. So, lots of words, and then we'll get to a picture and I'll go over it again. After the electrons from photosystem 2 have moved through the electron transport chain, they are accepted by photosystem 1. I know that sounds backwards, and on behalf of scientists everywhere, I apologize to you for that. We discovered photosystem 1 first, and then photosystem 2. When the photosystem absorbs energy from the sun, its chlorophyll loses an electron, and the electron goes through a second electron transport chain.